you can see something again. So humus and carbon. Capturing carbon through the growth of microorganisms and their transformation into necromass. Microbes are the planet's largest terrestrial stock of organic carbon and nitrogen. Bacteria splits every 20 minutes. Nematodes multiply 50 times to 500 times within 2 to 4 weeks. We already talked about the nematodes and how much bacteria they eat. Carbon cycle has much higher potential for sequestering carbon and is much quicker compared to grasses, trees, and other plant biomass. Carbon is king again. Humans are recognized as the single most productive input in sustainable agriculture. Humans are the only substance with the ability to hold every nutrient in the soil and increase nutrient uptake by up to 40% by themselves. Some of you that have seen some of the videos, this is a farmer back in 1958, I think it was, realized the importance of carbon. And so as a carbon source, he used dry ice. He dropped 1 pound cakes every 5 feet during the middle of the growing season. A small section of the field of dry ice yielded 211 bushels. Back then that was a lot, compared to 158 where he didn't. Carbon, is it important? This is simply photosynthesis, C6, 6 carbon dioxide, 12 water molecules equals C6H12O6 sugar, plus releasing oxygen, of course. This just talks about, this is the explanation of that little deal I just explained. So here's the problem. Organic carbon content in most soils is now 50 to 80% less than the original level. And that's no understatement.
That's okay. Well, they want to talk about stem cells.
So those of you that have runoff issues, we have a lot of that in Northwest Iowa where I'm from.
It's pretty cool.
The soils can absorb water. They can also hold capacity to hold it. And both of these comes down to carbon. 1% increase in soil carbon increases capacity to hold 23,000 gallons or an inch of
He had
water.
diagnosed me with COPD.
Get your soil tested for carbon. This was a test done.
And I got
These are what they call the Morrow plots in Illinois. Goes back to
And then I had to take a
1870,
reading test.
where they did a variety of things. They put corn, oats, hay in rotation. They put corn, oats,
They took
from 1885 to 1953, and then corn and soybeans
I'm taking
to the
one
present time.
blood thinner.
And they had to use corn. So this is what happened to the soil organic carbon steadily declined through those years. Now this was another one, the Sanborn Fields in Missouri, where they had wheat with 6 tons of manure per year. That's these right here. They started out with 4% organic matter in 1888. They put on corn with 6 tons of manure a year, continuous wheat and continuous corn. And these little symbols show the difference on these, and you can see that even with manure, the organic carbon levels went down. CO2 has a higher potential than nitrogen or any other mineral. If we look at those other graphs, this shows, for example, these are your N, P, and K and all your nutrient requirements right here for a 300 bushel corn yield. Okay? That's what those red things are down there. The blue line is the amount of carbon dioxide that it takes
Companies.
to meet the same requirements to grow 300 bushel per month.
Flies are
Just
off.
trying to make a point.
100.
So the Haney test is one of the ways you can check for carbon, soil health, and your C/N ratio. Like Dan pointed out, you can have a bunch of carbon, but if your C/N ratio is out of whack, you still haven't accomplished anything. It's all about the balance. This is an example of a Haney test, for example. The question was asked about CO2 bursts. Here's one at 158. The C/N ratios are down here. This is the organic carbon, this is the organic nitrogen. And here's the C/N ratio of 16.5. You probably can't see it very well. It also breaks the nutrients down into water soluble, and this test here is acid reduction, just like the exudates would do in the root zone of a plant. And this is your total number here.
Back up,
Yes,
go back there. You guys were talking about iron and aluminum earlier.
hang on.
The Haney test is very effective to see how much active aluminum you got.
Yep,
It's on the H3 extracted, right at the bottom, you got iron and aluminum.
right here.
You get them numbers above 200, look out.
217.
That's high.
Yep. Yep.
And how's that correlate?
What happens when
What's
it
his
gets
carbon
high?
number? What's his carbon number?
Pardon me?
What's the carbon?
The organic carbon is 336.
Okay, that's good carbon. So this is why it's important to keep your calciums up, especially your soluble calcium, so you keep that aluminum in check.
Yes, I agree. That field had a quart of biomentamon in the spring, poured on in the fall for 2.5 years. This was the beginning. This was in the 3rd year. That soil, when we tested it, which is not normal, when we tested it between the row and in furrow, was identical. I could show you. This was okay. Oh no, this was in furrow. So this had been soybeans, but between the row numbers look identical to this. And once you start testing this, you'll understand what I mean. But there was a big difference. Okay. Again, this is the test. If you guys want to take a picture of it, you can do it. This is the same one. It's Church. That's bacterial to fungal. Now here's the fungal number, or the total number up here. The bacterial to fungal here, 34% fungal, 66 bacterial. And this guy was using the ABC in furrow too. Okay, this is between the row, 25% fungal, 75% bacterial. Humus, we talked about that, humus functions. Okay, before we get into earth optics, there were a couple of questions.
I wasn't here this morning, but what is, with our prairie soil when we started, what was the fungal to bacterial ratio there?
50-50.
50-50, okay.
Now it's 90-10
Yeah.
at best. More or less. We're running into it. If you I've got guys that put in, I've got bacterial fungal tests on guys with 3 different insecticides. And this one guy that used a double rate insecticide in furrow because he had corn on corn every year. And he was using the biology in furrow. his numbers in furrow were lower, his fungal numbers in furrow and total numbers, bacterial and fungal, were lower in furrow than between the rows. Between the rows, his total number of biology in the soil was higher than in furrow. And the more these guys backed off, the one guy was using insecticide over the top at half rate, and in fact, it was the guy that I just had those numbers up for. But what also happened went along with that, invariably the yield went up as fungal numbers went up. With those, and I had one other guy who used a different kind of insecticide, and he was in the middle, kind of. So, but there were some questions here over noon hour.
Well, connecting the dots, so like we're starting to see insect die off, pollinator bees are dying off. Is there a correlation between soil health and what we're seeing here in the environment?
You know, the timing is interesting because this morning I just got one on my phone. There's been a study done where they're comparing both fish and bees, for example, on the effects of insecticide, fungicide, herbicides, and what is happening to them. And of course, the effects are negative, but it's affecting behavior to the point where with bees, as far as the brood and all that stuff, and how they're handling honey and all that, it's messing it all up. So it's messing up the whole system. They're not talking about death, but you know, when a bee doesn't do things right in the hive, something's going to happen. So there was a big study done. Now, of course, that was over. But they're doing more testing on it as well.
I'm going to add my own observation. Sandy and I grew up with crickets in a house up in Clayton County, an old farmhouse. Same way in our house where we're at now, in Delaware County, we have seen 2 crickets this fall. That's it. I've seen 4 caterpillars, I've seen 2 monarchs. I grew up with gobs of yellow butterflies and moths, whatever you want to call them. There's none of those anymore. A few white ones. I think 2 granddaddy longlegs. No bumblebees. And a few wasps. Take it for whatever it's worth.
I know.
I mean, we used to always have to be washing our windshield every day with blood splatter. In the front of your car was blood splatter. I mean, we'd wash our headlights and get so clogged.
Yeah.
There isn't anything.
No. You have to clean out your radiator too.
Mm-hmm.
So, where the plants grow, photosynthesis creates sugars in the plant, the biology sugars,
Yes.
and then the sugars that feeds the biology. The biology goes out to the mycorrhizae, it comes back and we're feeding. So it sounds like there's a language in the soil. I don't know if that makes sense.
Dan, you want to talk about that?
My name's Mike.
Mike, sorry.
All right, Vernon. We just got introduced, so I'll let you off the hook.
We've talked on the phone many times a number of years ago. Sorry.
Okay, there is a language, it's called plant hormones. The hormones have different signals, plant signal biology, to go to work. If you want to study it in detail, you can go to Dr. Wright's website out of Rutgers University.
Or Jim White,
Or,
Professor James White out of
yeah, well, that's what I meant, thank you, Mark. James White. I keep getting his last name. But he has noticed how bacteria actually go inside the root, and the plant strips the outside membrane off to get the nutrient it needs, then kicks the bacteria back out and says, go do it again. And then you have different fungi that live inside the root. Those are called endos. That's what row crops use mostly. Your forestry shrubs use mostly ectos. They live outside of the root. And there's a difference between the species and how they feed. But it's all about the plant signaling, and then you gotta have the food for them to go. Now, plants can put out a signal, but if they don't have the food for them to go, your soil biology goes on strike. Because they don't have the energy, and that's where the carbon comes in. And the amino acids and the lipids. So there's proteins, there's fats, and there's carbohydrates or lipids, they call them, that affect how the biology grows. And that's all about your soil health. So there's several tests you can do in your soils if you want to study how your signaling goes. The favorite one I got is called a PLFA. That measures your bacteria, protozoa, and plus and negative gram bacteria. If you want to get more specific, you can get into the DNA testing. But yes, your signals are important, and there's a lot of language. Even trees talk to each other, even from a distance.
Even when plant senses a predator attacking the roots, it'll send out a signal, mostly to the fungal, to attack it. And there's good and there's bad predators in the soil. Nematodes is a good example. There's good and bad nematodes. And if the plant senses something, it will send a signal out. And it will ask the good biology to attack the bad. They've done all kinds of stuff with that. And believe it or not, I have a book from the guy that did the original, for the government, made the first lie detector. And he actually attached the lie detector to the plants. And he could be in a different room and think to himself, I'm going to go and chop that thing off. And it would show up on the lie detector. You know, we have no idea the signals and what's happening between us and nature. Or if we hate something, or if we love something. I have a little saying on my wall, I have for years. What you think will happen probably will. So there's all kinds of signaling that goes on chemically in the soil with plants and biology. You have to remember, when the plant sends exudates down to the biology, and most of it's fungal, and asks for iron or asks for phosphorus, if it doesn't bring it back, it doesn't get fed. So there's a symbiotic relationship there that is extremely important. And we've only begun to, the research all been done. The chemical companies are not going to talk about this. And we just need to bring balance back, not this. And it all comes down to these two stupid little models. That's what your farming's about, believe it or not. That's it. And if you don't get those balanced, it's going to continue to cost you money, and then things will just get worse and worse. You'll see. Remember I said that.
Okay.
So, yes.
You're talking in furrow with your ABC. How are we doing on the hayfields, alfalfa?
Oh yeah, Clint.
Foliar, but on to spray.
Is it gonna, you were saying it's better in furrow though, so is it gonna get us our effectiveness?
This
Or?
responds both ways. Pastures too.
Okay, what stage would you say?
As long as the plant's green and growing.
This would be a ground
You don't want to spray it right before you cut it,
Right.
because obviously you ain't gonna have time to move as much as we harvest off of it. So I like to do mine as soon as it gets 4 or 5 inches tall.
Just get a good ground cover so it,
Good ground cover. you'll get some of the soil covered. And move systemically through the plant, so they will go down into the roots.
What kind of relative feed quality increase do you see?
I haven't done an increase on mine. Has anybody ever sent samples? I know we've had tonnage increases, but I've never did feed quality.
Let me answer part of that as well. So 3 years ago, we have, up by Brookings, South Dakota, young guy farms with his dad. They raise organic beef and sell it, okay? So they've got pasture, and they've got grassland, and they've got crops. On their grassland, brome grass, they've been same pasture for years, and they've been getting about 84, 85 bales per, big round bales per acre, or on that, not per acre, on that field for, I don't know, 20 years. He started spraying just this, 16 ounces on per acre. And the first year, and he didn't do that until midsummer, I think it was. First year, he went to 104 bales instead of 84.
Right,
Found it hard to believe.
right.
So he did it again the next year, and it went to, I think it was 130 some. He did the same thing on his pasture ground where he grazes cattle. And he's smart. He free choices mineral to his cows. And they normally would eat 2 to 3 ounces per head per day on the average when they were grazing. When he sprayed this on, they went down to about nothing.
Right.
So it changed the nutrient value. They were getting more out of the ground. They changed nothing else. So hopefully in part that answers your question.
Well, we grow dairy quality hay and we foliar after each cutting.
Good.
I've seen some 335 on RFQs. Now, I didn't believe that, so I sent it back in. It came back at 514. No way, the lab had to have lied on that. But it just blew me out of the water.
Yeah.
But a lot of it had to do with going in and spraying foliar after each time.
Right.
And you can see the composition of what's going on inside change tremendously.
Yeah.
And it has to, because some of the alfalfa, how many leaves guys typically see on a stem? 3?
Yeah.
Well, we've got 5s and 7s now.
Yeah.
So obviously, the plant likes what we're doing.
It's all about, you know, it's just like with corn, you get heavier test weight if you get the micronutrients. It's pretty simple.
How much of that are you spraying per acre, and why don't you use the A and the C?
You
Well-
can, if you could put it on there,
Yeah.
if you wanted to. We just do the biology because it's $3 an acre to a customer. It's very low, it don't take a lot.
Is that about right?
Was it 16 ounces up there?
Yeah, go ahead.
Every cutting,
or are you doing one cutting?
What's that?
You doing it every cutting, or are you?
I did it, this year I did it on the first cutting. I'm selling all my hay. I get out of the cattle business to go help grow in here, so my hay program has changed. I will tell you this, this was the second year of this cutting, or this alfalfa. Last year was new seeding. And I sold first and second cutting weight out in the field, and we're at 5 tons. The average tonnage in Nebraska in my area is 6. And third cutting and fourth cutting are sitting, there's 230 some bales. So you figure 1,600 pounds of bale, we're going to be pushing 7. So, which is huge. And I don't know, I was telling somebody earlier, the guys that bought it have hay too, and they have cows. And they mowed their hay first, and then they come over and mowed ours down. And he called me after about 20 minutes being out there. I said, "You having problems?" He goes, "No." And he goes, what did you put on your alfalfa? And I said, what are you talking about? He goes, well, he says, I'm having a lot harder trouble cutting yours, I gotta go gear slower. He said, go 440. I said, oh, really? And he goes, yeah. I said, the only thing I've done on there is I put one shot of biological on before first cutting. And he goes, I need to get me some for that, for mine. But we had so much moisture. Second cutting, if you timed it just right, get it on. Third cutting was a disaster. It was just humid every day, hay never dried. Probably the worst hay I got sitting there. I got some RFB, 90 on dairy quality hay. That's how long it sat out there.
Yeah, but you know there is actually no test for RFD.
Yeah, that's
It's a
just the
lab.
way, yeah.
So I mean, when people start saying that, throw that number around, you're talking blue sky.
Right. I know the industry's changed, but that's just kind of what everybody goes by, and our
I know.
RFQ is definitely.
Everybody does, but they balance the rations out, you know, but there's no actual test for it.
Okay. I don't know
It's-
anything
it's a
about
lab
testing hay. so-
Technicians' evaluation, what they think it is.
Okay. But anyway, so, yeah, he was sold, he's gonna spray his next spring. So, he didn't even cut it for me, like, hey, there's so much hay around, he let's let it grow. Everybody just got sick of it.
Speaking of quality, you know, not just alfalfa, but all crops. I know you don't get paid for quality, but my real job for 30 years was livestock nutrition. And I balanced enough dairy rations. I worked with about 50 herds at one time. And when you get the difference from different fields, and then you got to balance, you know, why is that happening? And of course, I've always got to ask the question. And that got me into what I'm doing today. You know, why? I worked for a co-op. Not for long, but I did. And that got me started in this today. But one thing before Clint let you know about Earth Optics, on the left-hand side, if you could throw the lights on for a second here. There's just one on SDS, SDS root of all evil in soybean fields. It's a big problem. Again, we'll know a little bit more on DNA testing and how much we're doing with that, because we use the ABC and furrow on corn and soybeans. But on the back side of that, this came from a producer up in South Dakota. And it's got the first one on top there on the left under Myers, ABC with bifenthrin, and ABC with no bifenthrin. There was a little bit of difference there, about 8 bushel, I think, on that corn. And then Relay. How many of you are familiar with Relay? It's I think it's a Dowdy product that they market. They talk about it all the time. With Relay in furrow, 213, with Biomentum in furrow, 219. Relay in furrow, below that, Biomentum in furrow, there was 5 bushel. And then on the next page, where it says farms, it's got another one with bifenthrin and with no bifenthrin. That just shows you the negative effect that some of this stuff has on biology in your soil. And this was with corn, the Biomentum in furrow. So, just saying, it's all there.
Bifenthrin's an insecticide, you didn't know.
Yeah, how many of you guys use that?
It's used almost everywhere in our area.
Yeah.
It's usually applied with fungicide. Cheap.
And then I threw this little, that's the next one in there. This shows the cumulative solar radiation in this area in 2023. Over here where the red lines start, that is about R1 and R2 and then grain fill. in 23 and 24 in this area, that line was going up. This past year, it was going down. And everybody, just about everybody out our way realized a decrease in yield this year, and that may be part of it. I'm not gonna say that was the only thing, but this stuff all adds up. Okay, with that, I'm gonna turn it over to Clint. And he's gonna explain a little bit to you about Earth Optics and what kind of a management tool that is. So, you get this.
I'm gonna hit the lights here. Yep. All right, so my wife and I have been doing soil sampling for about 10 years now. She's actually out right now doing it back at home. We used to just do 2.5 acre nutrients when we started, and then we got into the biological side of things. And then we found out about this company. It's called Pattern Ag when we first started. They've actually merged with another company, and now they're called Earth Optics because we have a lot of different stuff we do besides just biology. We also do high resolution nutrient maps and all that, so you can see exactly where all your nutrients are. Just going to show you what some of the results are. And some of these, I'm going to point out a few things in my fields that we're seeing and what might be causing it. And this is what we encourage people to do. Now, we can sample out here, or I'm sure there's a sampler out here that you guys know that does it. So if it's something you're interested in, just get a hold of me and I can find somebody out here, or I can send somebody out here to sample it and work with you if you want to work with us. So it doesn't make me any difference what you do, just as long as you guys can take advantage of it if you want to. So. Yeah, so basically this shows you all of your things we can do. We do a true nutrient, which is hard to see, but it's a scan. So instead of pulling your nutrients every 2.5 acres, what we're doing is we're going out every 5 to 10 acres to a point. We try to go to 5, but not everybody's field is square, so it doesn't work out that way. But what we do is we go and pull those nutrients at a point, then we have a big scanner on the front of our rig, and it scans it, so then we take the dirt, send it to the lab, and get it analyzed for nutrients. And it can sense those nutrients in the soil with AI and all this technology that's way above my head. And it'll actually make a 10 meter by 10 meter accuracy map of your nutrients. Now, there's nothing in the industry right now that can spread fertilizer that fine, but you can narrow it down from 2.5 acre grids. Like, I have an 80-foot spreader when I do my fertilizer, so I shrink that down to 80 by 80, so it kind of matches my spreader. The reason behind that is 2.5 acres is a pretty big area. Not all of that 2.5 acres needs what that soil test says, so we're able to actually break it down, but that whole square will get spread what the majority of that square needs. But now if we get more accuracy, we can cut half of that square off and then catch some of the next one where it overlaps onto the next one. So you get a little better accuracy. You can do a strip till, anything you use to put your fertility in, you actually do it with liquid. You can make a liquid prescription and run it through your planter on that. So we're also doing what we call True Bio, and that is, we pull samples in zones. It's a completely different sample than the nutrients. So we like to go 20 acre zones. We find that's probably accurate enough to give you guys an idea of what's going on out there. In that zone, we pull between 12 to 16 samples as we're driving across it. Also, on the scanning part, we're driving every 60 foot in your field scanning. So we're doing a good coverage. It'll actually scan farther, but they like the accuracy of 60 foot because that seems to give better accuracy for the map. So while we're doing that, we'll keep the zones separate. As we drive through them, we'll grab samples across the zones and put those in a different container for the biology test because they're two completely different samples. And then we'll analyze that biology test and we'll go through what's on that here in just a little bit. Another thing we do is True Till, which is just a compaction test. I don't push it. It's very expensive. It's $5 an acre. If a guy's gonna till, he's probably just gonna till. If a guy does want to find a spot, yeah, we'll go out and do it, and he can just go work that spot if he wants. But you have to decide if it's worth spending the $5 an acre to go test it. The reason I don't push it is because I've been tillage for a long time, slowly trying to work my way away. And what I found out is I can go out and disc rip it in the fall, come back and test it again the next year, and it's just as hard as it was before. So you can't fix it. You can get by for a little while, but it's not a permanent fix. So now that I'm focusing my resources on trying to get the biology deeper into the soil instead of, just trying to tear it up. Because every time you tear it up, you disturb all your biology in your soil. Doesn't necessarily kill it, but it disturbs it. So, you got next, Rowan?
That's all.
You didn't print those other things off? Those aren't on there? Okay. Well, then we'll need lights. Okay. All right, so you have these sheets with the bar charts on it. Okay. On the back of the one with the dots on it. So backside of those ones with the dots. Flip it over on the backside. So so there's one with dots like this, and then there's one that's the solid squares. I don't know if I have that or not, but that's an actual corn rootworm test. Now the reason why there's one with zones and there's one with dots is because in 2023 when I sampled that, our standard procedure was to do the biology with the nutrient test. So they just took the dots, joined them together to make zones when they did the biology test. The next year, they made the zones that you see. So I tried to make the zones in the right shape to try to catch to see if we could fix that rootworm with the biology. Now, what we're doing in that test is we're actually testing the rootworm eggs. And it'll actually identify between northern and western, which I don't know what you guys face here, but we have northern and western back at home. And the biggest problem for us is the northern, because the northern rootworm has now adapted itself to not hatch during a soybean rotation. So even if you do corn, beans, corn, you could still have a rootworm problem after soybeans. And we have had terrible issues with that because people didn't know, and they just figured, oh, I planted beans, I'll put a double probe. Well, we go out and sample a field now. I had a customer, one half of his 80 acres had over 70 eggs on it after beans. Clicked on it, told us it was northern rootworm. 2 years extended diapause. So they would have hatched that year. So he either put insecticide on or he planted a spark stem. Something to help prevent that from taking effect. We had it do devastating. That year was bad. Luckily we caught his. He does sampling every year. I was actually on the phone in the parking lot when we got here this morning. So we have tons and tons of stuff we test for, and I'm just going to kind of go through it. This is a field of mine. It's dry land. It's actually been corn on corn every year since the late 80s, except for 3 seasons, because my dad ran a rock through the N6 linear combine back in the late 80s, and he said he'd never plant beans there again. But the last year it was soybeans. I did plant it in 2019, the soybeans, just to kind of get rid of the corn. And we had a couple rock scares, so I didn't do it again. So anyways, so if you look at the one that says corn, and then it says foliar, that's the foliar diseases that we test for corn. We have different crops, corn, soybeans, cotton, there's just a list of them. And if you go on the website, or if you want to see it later, I can show you later as well. But if you look This is the corn foliar on the back of that one with the dots. And the top is where my samples from 2023, that got cut off, and below it is 2024. Now, this was last fall before this last crop. I haven't got it sampled yet to see if we can change these numbers. But this is how we're gonna monitor to see if we can get the biology to build and get rid of some of these diseases and pests and all that stuff in our soil. Now, it's gonna take a couple of years of data to do it, but at least we have a way to say, hey, we're gaining or we're not. Everything's got a reason. So, and they did change some ranges. So if you look at the first one, gray leaf spot, it stayed the same, but 0.01 and 24 is a little bit riskier, so it's red instead of orange, I guess. Goss's wilt, northern corn leaf blight, tar spot. You can see in 2023, we didn't have a lot of tar spot. In 2024, last year is the first year we had tar spot, and it showed up, this is in the soil already. So you can see that it's there. Now, the advantage to that is you'll have more than just this. We'll have the zones broken down on your field, and you can go back and see what that test was. This is the field averages on these. And you can see which spots had the heaviest tar spot.
The dots, one of the dots.
Yeah, right. They'll have the dots or the zones, however you have it set up. And you can go see what spots are your highest. So then when you're having corn out there and you want to go wonder if you're starting to see some tar spot, instead of wondering where to go, ideally you would go to where it's highest in your soil to go start looking there, because then if you see it there, then more than likely it's coming on.
Question, did you happen to analyze the brown bump on the leaf to see what the level of manganese was in it?
I have not known.
See, my mentor is Professor Don Huber that worked at Fort Detrick on biological warfare agents. He's 90 years old and still sharp as a tack.
Okay.
Well, anyway, he and Michael McNeil and some other people I know were responsible for doing a lot of the trace mineral work on crops at our land-grant colleges, and published in 1965. And that's the same time they did our trace mineral work on livestock and on soils. And they said in the U.S. Midwest we have the best soil in the world, and we do, and we still do in a lot of respects. They said we can separate livestock farming from crop farming. They said this in 1965, and get by with it, just putting on NPK and maybe some sulfur and boron, and just forget these trace minerals. We'll get by with it for 40 to 50 years. So that's what they said in 1965, and what the press and our ag industry heard is we'll do it forever. And so regrettably, if you take 45 years, the average of 40 and 50, and add that on to 65, that's when you got sudden death syndrome in soybeans and Goss's wilt in corn, and now we got this tar spot in corn. And they're all, according to Huber and McNeil, and I agree with them, they're all abiotic diseases. The plant shares 40% of its genome with us, and they need those trace minerals in order to fight off disease. And the manganese level, that's manganese oxide, so it's Mn+3. It's like iron oxide, which is ferric oxide, Fe+3O. Hemoglobin, if you have Fe+3, we can't use it. The plant can't use manganese in +3 state. It needs the reduced, and it doesn't have the reduced because it's sucking the hind tit for zinc and sulfur and some of these other stuff, see, early. And so Bob Strait, regrettably, that was a disciple at Central Iowa Agronomy, now he died this past spring, so I don't have him to work with. I'm not an agronomist. But when he followed Huber's advice and used his seed and graphite seed treatment and then some foliars or whatever, they aren't using these fungicides and stuff. And it works. Because they're feeding the plant again, and they've got better soil health, and so it can fight it off.
There's a lot of guys that can get by without fungicide, and it's because they basically know how to treat it without it. And I've seen it, and I'm not smart enough to do it, but I'm willing to learn eventually. So keep moving here.
So, as a veterinarian, And this guy knows it too. You know this dude, don't you?
Oh, yeah.
Yeah.
We know each other.
You have found out, and this isn't good for your practice, 90% of disease issues in livestock is because of mineral deficiencies, isn't it?
I agree with that. Yeah, or what's sad now we got excess. Yeah.
Say that again, Art?
There's a fine line between deficiency and excess. So because of the glyphosate, now I'm really ticked off because I'm dealing with copper poisoning in dairy herds and that, because,
You say that for when you get up there.
yeah, I will. Yeah, we'll quit now. But anyway,
I
yeah,
know.
excesses are just as bad or worse than deficiencies.
And you see, that's what we're trying to correct
Yeah.
biologically. And that's why we do the tissue sampling. And it is so much fun to watch that. But, you know,
The interrelationships are more important than the absolute values.
every time,
Exactly.
every time.
That's that first word, balance.
Balance. Sorry, Clint.
For the sake of time, I'm not gonna go through every single disease, but you can kind of see them. But I'm gonna point out a few things that we happen to see here. There's one on here that says, crop protection, and it's got the soybean next to it. So 2023, we've been in a 5-year drought until the spring, June, when it finally started raining. We were severe drought. And so if you all found that page with the soybean cyst nematodes, the top is still 2023 and the bottom is 2024. Soybean cyst nematodes need a host to reproduce. This has been corn on corn for 4 or 5 years, since 2019, so 5 years, 4 years. How did we get soybean cyst nematode to increase? Anybody have an idea?
Find a new host.
Yep. What host do you think we used?
The weekend corn or the weekend grasses?
So what happens when it's drought? What else doesn't work when it's drought?
Herbs.
You're right, herbicides. Guess what's a host for soybean cyst nematode that I just found out last night at 10 o'clock?
Water.
Because I saw this and I'm like, wait a second. Because I was always told that soybean cyst nematode though it has to have a bean. Water hemp, #1 host weed for soybean cyst nematode. And it was bad, so they couldn't kill it. No moisture, couldn't kill it. Showed up on the test. You have no idea what's going on in your fields, what's all going on in the background. This test will show you that. This test, I run it for my customers, $16 an acre. The local co-op charges $12 to do 2.5 acre grits for nutrients. $4 more. Would you rather know this? Or would you rather just know your nutrients? I took this to the co-op. We sampled for a co-op as well. And it's a very funny thing because I go in there and I said, hey, this is a better program. We can get you tied in with it. If we do your sampling, we can do it with it. Offer it to your people. What do you think the first question he asked me was? Can we make our own recommendations for fertilizer off of it instead of using the university formulas? Nope. We don't do the sampling. This year, they're allowing them to do that, but I haven't told them about it yet because I don't think they need to know. Now, I will tell you this. We normally do sampling for them. We're usually swamped. We've done 4 fields for them this year. For the guys that we're doing this for, she's been gone for 2 weeks. Every week she's gone. I haven't seen my wife since Monday. She got home last night because I left and come here, so she could be home with the kids. And then she's gone this morning doing stuff close by. We don't sell fertilizer. Do not go to the co-op, the person selling you fertilizer, and get a fertilizer recommendation. They all have their own formulas. And one thing I noticed when I got off of my co-op fertilizer is my fertilizer rates to apply are not as high as they used to be, going off of the university data or formulas that are in this program. And yields have not dropped. and levels have not dropped. So just keep that in mind. I'm not saying all co-ops are evil, but they make money somehow. And there are a few guys that are good, that they want to sell you fertilizer. That's how they make money. But you can go through and look at all these. You can see the diseases we test for on soybeans. So as you go through them, you can see there's biofertility and things like that. So if you go to the soybeans, this is one I noticed last night too. Go to the soybeans and the biofertility. And you can see all the things we test for. And the mycorrhizal fungi is one I want to focus on. Now, what happened there, we had 2023 and then 2024, it went down. I still applied it. I did fall tillage, but it was dry. Gotta have moisture. So that showed up on the test. So you can monitor all that. The one thing that went up, and Drew and I didn't know what this was. We were talking about this on the way out here yesterday. Sidor 4 production. Anybody know what that is?
Sidor 4 loves iron.
It's measuring the ability for your ground to make iron available, basically is what it's doing. It's gone up. Okay, so you can go through all these little diseases, you can see all those problems. But the one thing that hardly anybody tests for, and I gotta see which one it's on. It's not that one. Go to the soybeans crop protection. Might not have been moisture that brought the AMF down. What competes with mycorrhizal fungi? Just about everything else. We also test for nematodes. Look at the nematode levels. They're mostly all on the right. We got super, super, super high nematodes. Mostly all red. So, what do you attack when you have problems with yields? You don't know. So you want to try to figure out which is the best way to go do it. This gives you a bigger picture. There's different things in here you guys can scope through and see if you see a difference. But the thing that I learned about it is, instead of just throwing something out there, you can kind of see what you need to go after to attack. And with the internet these days, you can look every single one of those things up and see what it is and see what its natural predators are, see what kind of products are to fix it. I don't want to take a lot more time on it, but I think you guys have the main idea of what we're testing for. And this list is twice as big this year as it was last year. This is a soil DNA analysis. It's not a respiration test. Respiration test does not identify things. Soil DNA test will identify everything in that soil that has a genome.
Well, let's tell you, and Berlin will know this, there are such things as beneficial nematodes.
Right.
You just kill them off. So this test tells you these nematodes that are built up are not your...
These are the ones that affect you.
Okay, not the one that would help you kill some of them.
Yeah, so they just added those nematodes this year. I haven't had, like, they added them like 4 weeks ago.
Okay.
So I haven't had much time except the car ride to go here. I harvested, got all that buttoned up, and now I'm helping Berlin on the road. So I don't know what classes of nematodes are all the good ones. I'm sure they can develop them and start identifying them. But the biggest thing is trying to get rid of the things that are hurting you.
And the sad thing is, see, if we keep relying on our chemistry, there's no way you're going to keep your beneficial nematodes because you're going to kill them when you kill these others.
Correct. There is a guy, and I don't know, I think he's from Pennsylvania or something. He calls himself a nematode guy. And supposedly they have cultured nematodes that you can put out there that attack the bad ones. Because apparently that's a good way to attack the bad ones, is to put a certain type of good one out there. I haven't, it's expensive, but supposedly they can last quite a few years. If you do a treatment, I don't know, it's like 50, 60 bucks an acre. So I haven't looked into that yet. But after seeing this on the car ride yesterday, that's my, I thought you had it on the slide, so it's kind of hard to show you on the papers. But I wasn't prepared to see all this, and I saw it last night. I was up till midnight trying to siphon it out and figure out which is which, but there's problems out there. I don't know what we're gonna do to fix it, but nematodes are definitely a problem. So, just want to let you guys know that this is what we're using to kind of measure we're gaining. We want to see if we can bring the AMF up. There's plant growth promoters. That's things like your good beneficial fungi and things like that besides your AMF, protozoa, things like that. So when you go on the program and you can pick each category, each one of these, and then if you click on it, it goes down and gives you a description what it is. Tells you things, ways you can fix it, things like that. But that's basically all I have right now. I'm hoping when we have a spring meeting, I can have better answers for you.
Question, have you done any studies on how your nematode population stacks up to your mycorrhizal fungi?
Have I what?
Say that again, please.
Have you done any studies with your population counts of nematodes compared to mycorrhizal fungi? Because typically fungi are a competitor or a
So
predator to nematodes.
Yes, we are.
So
there you go.
The
There's
problem
your
is,
answer.
the problem is this. Yeah, this is the first year we're getting started on it, and this is the first year they're doing bacterial fungal numbers as well. And this is not taken in furrow. We're going to be sending samples in to check them for in furrow, in between the row, and run all these tests again so that we can see if we're having, like you were talking about, whether it's a negative or a positive or if we're doing anything at all. Because that's very important.
So you're just getting started collecting data.
Yeah, on that. Yep.
like I said, this nematode test just showed up 3 weeks ago on here, right during harvest, when I was harvesting. I haven't had time to go through all the data yet. But the thing about Earth Optics is I've been doing it for 3 seasons now. And if they did a DNA test on your soil, all that data is collected from there, so when they can identify another genome to identify on there, or markers to identify on there, it'll go back. So when these nematodes weren't tested in 2023, we didn't have that test available. They went back and they got the data off the computer and they said, well, that's what that is. So now when you go back and look at your results, it's updated. Fungal to bacterial ratios was just added here right during harvest as well. That's on here too. Let's see here, that should be in soil health or biofertility. It's in soil health. So the way they do it is they do it parts per million, which I've never seen before. They're working on changing that into a more one-to-one type ratio. But if you actually go to where the results are, not the summary, you can figure out the percentage. And what did we figure out on that? Like 26%
Yeah.
in that field. So basically, you just take it times 2. And then it's just the first 2 digits, so that kind of gives you an idea of what it is. So that field's at 26%. Now, I will tell you this, I have fields that are down 5% fungal. And I had a test strip this year where I did 16 rows without the ABC and furrow, and I didn't put any foliar on it. And then, so I took the samples in furrow, and in between the rows, and I had like 4% in this one field, not this, but a different field, and then like in the furrow, I think I had like 5. It wasn't over 5%. It went over exactly 8 rows, where I put ABC, and I did the foliar pass, the one quart over top at B10. I wanted to do it sooner, but we ran out of product, so I had to wait until we had more available. Sampled it about 3-4 weeks after that, and those tests come back to 34%. 8 rows apart. So we know we can build fungal. Whether or not we can keep it there, that's what we gotta work on. We gotta make sure we don't do things that make it go away again. So that's why I'm starting to cut away from my tillage. I used to do a lot of deep tillage. Residue is a big problem. On my farm, I haven't done any fall tillage this year. We're doing salt digester, and we'll see by springtime. If it doesn't break down enough so I can no-till into it, then I guess we'll have to get the disc out to get by this year. But I'm trying to hold off as long as I can. Because if we don't let this stuff grow and get bigger, cover crops would have been great. I didn't have time for cover crops. It's expensive for me. But yeah, cover crops do help. But you get a short window to get them in, and where we're at, and you guys are probably the same weather as we are. It's very difficult. You can do it with the drone. I do have so I personally have a 20-acre tillage radish trial going on. Now I know it's not really a good cover crop. Everybody says, well, you gotta have multi-species and stuff. What I'm trying to do with that, so tillage radishes, they go down, get a bunch of nutrients, they freeze, they rot, and they leave pockets of nutrients and places for your roots to grow. Well, we all know the biology attaches onto the roots, and the roots go searching for the nutrients. So what I'm hoping to do is I'm using the tillage radishes to break the compaction and also make pockets for the biology to work its way down deeper in the soil. Then hopefully over time, I can build that biome deeper in the soil. I don't know if it's gonna work, but we're trying it. So next year, next spring, I'm going to keep it separate, of course, and then do different depths for the biology test to see if we can take that top layer of biology and the bottom layer, we can go different depths with our samplers and see if we can get it deeper and raise those numbers up in those deeper samples. Because right now, the deeper ones, there isn't any biology. So if we can start building that up, then that's going to be a good sign. Anybody got any questions? It's just basically soil sampling and getting more results is what it is. It's a very good tool. Again, like I said, if you want to work with somebody like us, that's fine. There's other guys in the area too that are just as good. All these guys are trained that do orthopedics classes. And just, this is another tool that's for you guys, just to help you guys figure out what's going on in your fields. So, I don't have time to sample everybody's field either, so, I mean, as long as you guys are doing as much testing as you need to figure out what's going on out there. So, I don't have much more for that, so.
All right, then we'll finish up here, and I know Art's got some interesting things to present, but just to sum up, biology is not the answer to everything, but it's a place to start. And if you want to get out of the rut you're at, you got to start somewhere. You know, the definition for insanity, right? So, again, these two are what your whole operation is about. Nothing alive on this planet lives without carbon. So carbon is extremely important, and especially available carbon. Okay, Bart, come on.
What's your smaller molecule up there? Hydrogen or oxygen?
Nitrogen.
Nitrogen, okay, gotcha.
Five
Sure, yeah.
carbon,
Yep,
one
nitrogen.
nitrogen.
Okay,
Bacteria.
gotcha.
Okay?
Yep.
All right, you're on, buddy.
Did you get my flash drive in there?
Ready to go?
That doesn't mean I don't know how to run.
Can you turn the lights down?
You guys are, I'm a veterinarian, but my partner, classmate, Brian and I were just south here. And we were, well, practicing for some reason long, because we started there in 1974, so this is the 51st year. And I ended up, let's see, in 2007. In 2005, we had a good-sized purebred Simmental farm. We worked for him for 43 years. He's dead and gone of cancer now. But at the 43 years we worked for him, he only tried to listen to his co-op, and he's right on the Coker River, down by Hopkins. And he went to Roundup, so he fed his cows. He planted Roundup, used Roundup for the first time in the glyphosate with it in 2005. And he fed his cows Roundup, corn silage, and Roundup grain instead of non-treated. And that following spring is the only year we worked for him where the year after that, where he sold fewer animals than what he exposed heifers and bulls to in 135 cow Simmental herd. And that year, we ended up having bulldog, little short-legged calves, a bunch of them born, abortions, and then they were born. They're manganese deficient. I'll show you a picture of them. They had to reach up to try to reach the udder, and short-legged and swell joints. And then manganese is real important for reproductive function. So these heifers and cows, when they have P ovaries, even when they're in good energy and protein balance, so they don't cycle. So his heifer conception rate went from 96 or better down to 83. And then we had navel ill and calf scours when we hadn't had that problem before. So that's the only reason we fed it. Well, we had it in that herd, and then I sent a bunch of gilts from some farrowing outfits where we had real good reproduction, and then they switched over to Roundup grain. And anyway, we butchered a bunch of animals locally so we could go into the locker plant and look at the tissues. But anyway, we butchered them before the holiday, before Christmas, and they had little itty bitty P ovaries. They were manganese deficient, sent in livers and confirmed it. So anyway, Dr. Don Huber became my mentor and friend in 2007 because I tried to, I live near Hopkins, and so that's where this guy is, and so we got Fayette, sandy, loam soils with a lot of slope, and they leach trace minerals way worse than black dirt or clay down around Ames, and they also manganese, cobalt, iron, zinc, copper, all those +2 cations are taken up better in acid soils. And we're over here with calcareous soils and that soils that leach. So it makes sense that Don and I would see this problem. Now they're seeing it around Ames, and they're seeing it and they're writing about it in Hordes Dairyman and stuff. It's kind of funny because they make it sound like it's something new. We live in my wife and I bought a 43.3 acre property near Hardscrabble down by Hopkins, and so it's half rough timber. The kids can jump off a 20-foot high line rock if they want into the creek. So all 4 of my kids, 2 boys and 2 girls, all grew up with a conservation ethic. So I was visiting with your fertilizer man here, Terry Frommel, about, we already mentioned, when my kids come back now in the summer, we all know, compared to 40 years ago, Rachel Carson, it's too silent. And heck, when you walked around my timber 40 years ago, on a wet day, you were going to get stung by a native honeybee. Shit, now you don't even see a bee. And I'm losing all sorts of ash trees, and this year, even though I got a few ash trees that are still alive, when I walk around my Art Dunham deer path, that's how I get my exercise when we get caught up in the afternoon. I walk up and down and around my square 40. I don't even see an ant scurrying across the path, or an emerald ash borer. And I'm not exaggerating, it's pathetic. I mean, yeah. We don't need any more research to tell us that in the last 40 years we've lost 50% of our insect population, both species, and then the songbirds go right along with it. So.
Hey, Art, just to interject here, we've got a guy up in Minnesota that uses the Biomentum to spray ash trees up there, and he doesn't lose ash trees. It stops the ash borer.
Is it heavier? Can we do that with us, Ron?
Both. It's more effective, foliar, but 80% of the roots are within 2 feet of the top. And if you dilute it in a bucket and pour it around the drip line, it'll react even faster. But we've got.
How do you get to the top of the tree?
You don't get to the top of the tree.
Drone, use a drone.
You can use a drone, but he gets about 30 feet up. But I'm telling you, if you have that on your ash trees, they will not die.
Every year, do it yearly.
Yep, he's had ash trees that are 2/3 of the leaves are gone, sprayed it on, and they bring them back to life.
See, we know we're getting oak blight now, and then people mention, well, all this fungicide spraying. Maybe when the crop went down a little bit in price this year, I'm not sure it was 90% of the people that put on 3 fungicides, but a year ago, they spent another $3 to put on insecticide. So my partner has got kids with families in Cedar Rapids and Des Moines. If he wants to see fireflies, he's got to go to the middle of the city when they aren't spraying for mosquitoes.
Yeah.
Yeah, and we know, now that we know about our gut microbiome and the importance of all this stuff, we got to work with Mother Nature. Yeah.
This guy in his hometown where he lives, he started, the guy that sprays for mosquitoes in the summertime, insecticides, he pours the Biomentum right in there with it. And that's how he sprays the ash trees too. So, anyway.
So here's a manganese deficient calf from one of our ex-clients that's now working for a big outfit, and so you see it, that was in 2020. So they weren't following our recommendations or whatever, and he has them as severe as some of them. Some of them are so, I lost my old computer, so I had to take a photo of the photo, so maybe it's not as good as it should be. Okay, anyway. Other trace mineral issues. Let's jump. I could talk forever. Here, Bruce, I think, came down to, this is what, 2 or 3 years ago, down at the client we kind of both give advice to, and he had copper poisoning then. Well, now he's going through it again on another batch of calves he bought. And it's, when you buy 400 Holsteins, steers, at 400 pounds, they've already been someplace where they were born from mothers that were overfed copper because they aren't using glyphosate. And see, it isn't that big a percentage of them. It might only be out of the 400 calves, it might be 30 of them that have this super bad anemia. And the yellow color's coming through. They're just totally yellow inside. So if they were an old cow, you'd think, oh, she's got anaplasmosis. But then the spleen's big, or she's got this new Asian longhorn tick, filaria. But copper poisoning causes an intravascular hemolysis, so just like the baby that's born yellow, it's got Icarus. And then regrettably, by the time we're seeing them, when they're feedlot size at 550 or 6, see they're breathing fast, sure they get pneumonia because they're so anemic, they got next to no blood. By the time we see them, this calf, it's hard to see, but I opened up its cecum, and that's black digested blood in the cecum. They start running out of clotting factors, so then they get extravascular hemolysis too. So when you send the tissue down to Ames argues and says, well, that can't, they did all their work, see, on acute copper poisoning, when they die real fast because you make a one-time mistake. But when it's chronic like this, they're trying to tell me if normal copper on a wet liver basis at Ames is 25 to 100, they're trying to tell me that you got to be 300 or 400 to kill them. Well, on some of these, I'm lucky to be over 100 because if copper isn't stored in the liver, the next most prevalent place it is in a mammal is in red blood cells. So see, if they're bleeding all the time, then that number goes down. So I hope you can, if you can't see it, copper on there. This is a aborted fetus from dairy heifers that were overfed copper. And Ames has been picked on because they had a plasma mass spectrograph. Actually, I leaned on them enough, they bought it in 2008. Regrettably, there aren't enough vets using it yet. And Steve Inslee was there as a toxicologist, and he got chewed on because they thought they'd never pay for it. But anyway, I sure use the thing, and I wish more people were using it. But you see copper on they used to say that a liver in a freshly dead animal, and if I'm second guessing a nutritionist, I like to have a calf that gets caught in the feed bunk or breaks a leg or something. See, I don't want to use something that's been sick for 3 weeks before I send it in. So I tried to send something in that's a cute cat. Well, here this thing was aborted, and they decided to, now that the University of Florida Vet School and Michigan State Vet School are both yakking and publishing about copper poisoning, now they decided they sort of had to believe me, and so this is on a dry basis. Well, before they said the liver is usually about 50% moisture, and so if you take your moisture out of there, if normal on wet liver is 25 to 100 parts per million, then on dry liver it ought to be 50 to 200. Instead, look what the numbers they came up with. The copper there, if you can see it, they say the normal range on a dry liver is 150 to 650. And what's my number? 540, or 6, my number, that's 6. My number is 645. Well, if we use their old rule, so I phoned them up and we had quite a conversation. And I told them, because actually the toxicologist up there, Scott Radka, that I know real well, was going to go someplace and talk about copper poisoning. And then he came up with this new test, and I told him, well, if you're going to try to convince people that it's happening, how are you going to do it when you change the rules? So this is kind of funny because now they've gone back to doing it the wet way, the way they were doing it before, and this is the only time I've seen my results like this. Cobalt. This is how I became a real good friend of Dr. Mike Sheridan from Steinbowl, Manitoba. And that guy, he's had to quit practice because he's had some strokes and had heart issues. But the guy talked at all these American Association of Swine Veterinary meetings, and he's constantly thinking. And anyway, he works in West Steinbowl, Manitoba, so he consulted for all these huge Hutterite colony pork outfits. So this one outfit, and I think it was in, I don't know if it was in Saskatchewan or Alberta, but anyway, farrow to finish, 64,000 head of hogs a year. And they were raising all their own feed. And he started running into squatter pigs. Of course, they're in kilograms, but I converted over to pounds, so they're about 150 to 200 pounds. These pigs would go down on the ass end and start dragging they were paralyzed, dragging their rear end around. So Sheridan, at the time, they're doing a lot of things right because they aren't feeding corn and soybean meal, they're feeding small grains. But they were doing a lot of things right because guess what their weaned finished death loss was 2.7%. And yet he started having 5 to 6% of these pigs would go down in the ass. So he cut into them with a skill saw and sent the spinal column into a vet school, and they said that's demyelinization. Due to myelin is what surrounds your nerves. It's like having electric wires that have adequate insulation around them. And if you don't have enough myelin, things don't work. So it doesn't have to be the spinal cord. Sally Packalock that wrote the book, Could It Be B12? She says 40% of Americans are low on B12 now. And I agree with it, they probably are. So some of this, your arm going to sleep or your leg not working right, it goes back to this. So anyway, what Sheridan did, after sending it to 2 vet schools that told him it was B12 deficiency, he sent it to a human medical school, and they told him the same thing. So then first he put 5X, the amount of vitamin B12 in the feed. It doesn't cost all that much yet. They used to make it in sheep in the rumen. So your ruminants are supposed to be able to, with the bugs in the rumen, to manufacture it. Now they ferment it. But anyway, he put in 5X, then he put in 10X. And he could only drop it down to 3 and 4%. He didn't get rid of it. So he posted back now on the AASB, it's worthless because if you're on that thing, you got all we got left pretty much now is big companies and then nobody will share anything. But back then you could actually talk to each other. And so he left his phone number and he wanted some help to try to figure this out. Well, I'd already phoned up Don Huber in 2007 when Ames went and I knew about glyphosate chelating cobalt. And so I phoned up Mike and I said, is there any chance this Hutterite colony, they have big families like the Amish and all sorts of young labor. I said, do they quit windrowing their small grains if they weren't quite right and then spray them dead with glyphosate? And I still remember on the other end of the phone, there's a big quiet spell and then a bunch of swear words and looked at me, I'm a slime bed. I don't know anything about that, Art. And so I told him, you better check it out. So he checked it out and sure as heck, that's exactly, they started doing that to all the feed they're feeding their sows and pigs. They started doing that about a year ahead of that. And to fix the problem, the 10X add more vitamins didn't do it. But they just dropped that out of the procedure, and then they went on fine. So what am I seeing? You see the next thing, anemic pigs. So in the last couple of years, as you can find cobalt there, the normal cobalt on a wet liver on a pig is supposed to be 0.25 to 0.5. Look at this. Ames still just writes low by it. Well, they didn't even write low by it. 0.005. That's 1/100th of the top end of the range. 0.009. That was in 2023. And these pigs are taken out of a double stock nursery and sent to our client. He phones me up on a Friday night. They just delivered them and 2 of them were dead on the truck. And he says, Art, I think they sent me pigs with PRRS. And I said, well, come out tomorrow morning and we'll post whatever else dies and the 2 that died on the trailer. So they'd only been on his farm for 16 hours. And so I ended up posting 9 of them, and then I made sure I took 2 pigs that were full of grain yet and that didn't have bad PRRS lungs yet. They looked normal. And that's the sort of values I got. And then if you go down to molybdenum is supposed to be 0.6 to 1.9, 0.53, 0.68. Well, molybdenum is part of the pituitary protein and vitamin B9. Well, you got to have B9 and B12 to make red blood cells. I think, where's the iron on this one? These they called the iron low. So here, whoever took care of these pigs, well, they were 45, 50 pounds, so I better not say anything. But a lot of times, years ago, we had all this hired help and stuff, and sometimes they wouldn't give iron shots. But what I'm seeing on the pigs coming in now, I'll show you 2 more of these, the iron's actually coming back high. And yet you can look at the pigs, you don't even need to do a hemoglobin. They're white pigs. Hell, a white pig still ought to have some pink color. You can look at them and you can tell they're anemic right when they let them off the trailer. So yeah, these were out of the double stock nursery too. What do we got for cobalt? 0.25 to 0.5 is normal, and they're 0.004. So that pig is. And molybdenum, supposed to be 0.6 to 1.9, and it's 0.5. He's going to be anemic, even if he doesn't have mange and everything else working on him. And here's the pigs I just got, or just went through this not even a month ago. So I'm talking to their nutritionist. And I phoned him 2 or 3 times because he said, 2 weeks to go over this, and he promised he'd get back to me, and then, Art, I think you're on to something, but I haven't heard anything from him, because I don't think he knows how to fix it. Here you go. Here's one page. I sent in 2 letters. One's .005, the other one .006. And look at the molybdenum. One of them's .33, and the other one's just a little bit above the .6. Oh, and these pigs were on the farm, our farm, for 3 weeks. They came in at 3 to 4 weeks old, and then they've been on our farm for 3 weeks. We're feeding them 1/2 pound of sodium molybdenate. That's 2,000 parts per million. So they've had that to eat for 3 weeks. So what do you think those molybdenum numbers would be if we hadn't put in that 1/2 pound to try to fix it? And then look at his iron. I think they gave iron shots. It's supposed to be 100 to 200, and they're 246 to 273. His copper's a little low. There's a lot of copper in red blood cells. So sometimes, it used to be when we had indoor-outdoor pigs, you gave them copper sulfate to follow up your water treatment, and then somebody is leaving it in longer than 3 weeks, and then you had copper poisoning. Oh, and I'll keep going here, but down below, Feedstuffs magazine today, I get it electronically, of course, and looked at it, and of course they're talking about all the shit we get from China. All our amino acids, pretty much, all our vitamins and stuff. Although vitamin A, I found out after seeing this result, I looked it up, we get 50-some% of our vitamin A for humans and livestock from India, and only 20-some% from China, and then it's only in the 18 or 17% from here. But anyway, some of it's no good when it comes, but they didn't give a normal range here, but for A, this 18 and 16 is supposed to be about 3 times that. And the vitamin E, I think, the vitamin E in this case was low too. So, what's interesting about this, of course, I immediately asked the fella, he gets 2,400 pigs every 2 months. I said, you didn't happen to, there's a whole bunch of little things we keep doing wrong. I said, you didn't happen to buy premix or have premix from your previous bunch of pigs, and then, oh no, I always order it fresh. So that sounded good. Well then 3 days after getting this back, I'm at a dairy client's. And I'm looking at heifers. They had me look at these coffin heifers, and they're in a building that has next to no wood in it, just the posts and otherwise no wood. And we haven't had ringworm problems for at least 2 or 3 years, and that's only occasional threat. So I look at these dairy heifers, and now we aren't having trouble with pneumonia because they aren't overstocked because they've been selling all the bulls in this 170 cow herd. Well, the calves in the middle, the real young calves are slick as moles, and the older ones are slick as moles, and the ones in between, some of them lost all their hair because of ringworm, and then they're coughing. So lo and behold, my hog client is getting these premix mixed at the same plant where they're getting their dairy feed. And see, as we get less livestock people, they don't want to be mixing premixes all the time, and so they want you to buy at least a 3-ton batch, and then once you mix your vitamin in with your mineral, you really better feed it up right now, because vitamins are antioxidants, and if you don't want it sitting around, even if you got it in it. I went and looked at it, and they're sure shooting that was the problem. In the center there, they'd had that stuff for almost 3 months. And you know, you want to feed it up in a month. Yeah, that's something you don't want to order ahead. The International Dairy Consultant, Earl Walseth, gave, and this is something for years, I didn't give B12 to a cow to stimulate appetite like I gave to a pig, because we used to say cattle and ruminants with the bugs in them, they could make all the B12 they needed and all the B vitamins. Now, not so much. Well, Earl Walseth, he gave, and he's retired now, but this is 10 years ago or whatever, he gave 7.5 cc's of the 250 cc bottle stuff we have twice a day to fresh Holstein cows for 2 to 3 weeks. He got 5 pounds more milk per day through the whole lactation on less feed. And I challenged him at the time he was doing it. I said, Earl, he was out of Washington State working with all these big herds, and he had a nutrition degree, and we got along good for some reason. Anyway, I asked him, would this work in a herd where you're feeding non-GMO or non-traded hybrids? He didn't know because he wasn't working for anybody like that. And again, see, if we never do the science and just refuse to look at stuff, well, then we can't possibly see it, especially when we're getting all our money from the people that still push it. And regrettably, in the book where I wrote a chapter and Huber did, I got it sitting back here, I knew nothing about glufosinate, and I just talked to Don the other day too to get educated on it, but glufosinate, is these pig cobalts again. I've been interested in vitamin B12 for years. Well, it's only been in the last 3 or 4 years where I have it in absolute shitter, where I'm 1/50th to 1/100th of where I ought to be. Before that, I was just maybe half a unit under the bottom end of the range. And I think what's happening is we're fighting this water hemp, and a lot of people, the glyphosate isn't working anymore, so they're switching over to Liberty, which is glufosinate, and they're spraying the hell out of their beans, 2 and 3 times right after bloom. And if Hubert sent me the diagram, I got it back here in the book, I should show it to people so they can take a picture of it. Glyphosate is a synthetic glycine. It's a man-made synthetic glycine. That's an amino acid with no side chains on it. And glyphosate has one little methyl group coming out of it. And glycine is super important because it lets proteins bend, and they're so long. So if they're in the cell wall, or they're an enzyme, or if they're in your collagen in your joint, it lets you bend. In industry, when we approved glyphosate, Monsanto actually admitted that when an animal eats feed with glyphosate residues in it, 68% to 66% goes right out the butt. And that's one reason we like to chelate our trace minerals, because it's harder for it to bind to them and take it out the butt. And then what they absorb is pissed off via the urine. And Monsanto had to admit, nobody thought about it, but they had to admit that there's a real small amount of it that never gets pissed off. So some people are theorizing, and nobody knows, but they're wondering, we got kids, we're pushing athleticism on them earlier and all this weightlifting, but we got a hell of a lot of joints going to hell in young people, and some people think it's because you got some of these stupid glycines with there's no way for your metabolic system to break that down. If it's in there, it's in there. Botulism. The botulism we're looking at now is toxico-infectious botulism. That's a big word. What that means is, when we think of botulism, we think of somebody not running their canner long enough and having the toxin developed in that jar. Or we think about killing off a whole bunch of cattle where you ground up a coyote, put it in the TMR wagon and fed it to them, and so a whole bunch of the cows die all at once. And we've seen that, but only once. And it was when somebody bought, actually you know about it, Mitch.
Yeah, know about it.
Yeah, you sold them the hay. And it had some dead woodchucks in it. So it killed their farm dog, and that gave us the answer. And then they sold, that's before I had to do a lot of phoning at night and stuff, because when the cows went down, they got mad at Don and I because we treated them for what we thought was lactational milk fever, and they didn't get up. So then instead of calling us back when the cows went down, they sold them to the Amish and they butchered them to eat them.
Oh, no.
Well, thank God, botulism toxin. That was before mad cow disease, see, so you could sell down cow. So I phoned up California and all over. And I found out, well, botulism neurotoxin is supposed to be temperature sensitive. So if you truly cook the meat boiling for 10 minutes or 20 minutes, you're going to kill it. And this ultra fast high temperature pasteurization is supposed to kill it, but you know in the last 2 or 3 days we've been hearing about botulism in a baby formula, where maybe they didn't have it heated up quite as much. Well, anyway, regrettably, the way to diagnose botulism is you're supposed to send it to the New Bolton Center in Pennsylvania. They're a veterinary diagnostic lab that's supposed to diagnose it, and you're supposed to take out contents from one of these dead animals, send it in, and they inject it into mice, and then the mice are supposed to die. Well, regrettably, mice aren't as sensitive to the toxin as birds, humans, horses, all species of livestock. They aren't as sensitive. So when you have Clostridium butyricum, it makes butyric acid that smells like rancid butter. I'm sure you guys, I got a sample in my truck if somebody wants to smell it. Anyway, if you make corn silage or haylage, and it doesn't have to be Roundup ready, if you make it a little too wet, that instead of getting the lactic acid that you want to preserve your silage, you get butyric acid from Clostridium botulinum. And then that Clostridium botulinum that made that keeps growing in the cow when you feed it. It isn't great feed. They don't eat as much, they don't produce as much, but they don't die unless you're a Roundup user. And if you got Roundup corn silage, Roundup grain, I guarantee you, you're going to be over 0.1 part per million, and that's going to knock out Enterococcus faecium and Enterococcus faecalis. And somehow now we know about the gut microbiome, and those bugs produce a fermentation product that keeps the Clostridium butyricum from making the toxin. And Clostridium novidei can do it, Clostridium hemolyticum can do it. And I learned all this from Dr. Monica Kruger at an International Food Safety Conference that I attended because of Huber in 2014 in China. And she taught me more in broken English, I don't know any German, when we ate breakfast together before we got on our planes to fly home. I learned more from her in 20 minutes than I've learned from anybody here since. And then what's really sad, my ISU has some excuses on this one. Because after 9/11, we said that botulism is a select agent. So they won't let any land-grant college or anybody do any research on it. You got to be a biosecurity, you got to be the new lab that's a Kansas State or a military lab to do any research on it. And it's all over. Monica Kruger knew way back when. Monsanto knew nothing about her. She was before the Berlin Wall came down, she was in Leipzig, Germany. To this day, now she's retired and she's getting some dementia, regrettably. But she was one of the world's experts on sudden infant death syndrome and crib death, which isn't always botulism, but sometimes it is. And she worked at their human health hospital on that, and then she was a veterinary instructor at the University of Leipzig. She had a PhD in mycology, that's the study of funguses, a PhD in microbiology, and a PhD in veterinary pathology. And I think she's one of the smartest vets I ever talked to. Anyway, she's so far ahead. Okay, so here's a herd bowl with this toxico-infectious botulism. And he ate a little bit more of the corn silage than the 35 beef cows he was with. So he was the only one that was unfortunate enough to get it. Well, that tongue is half bit off by him. And the first day when I should have done a video, I told the client to fill the water tank when I got there. This is the first day I saw him. Well, he filled the water tank, and the poor son of a buck he's trying to swallow. Well, you can't swallow, so it works on your smooth muscles. He's also staggering around. You don't know which way he's going to fall next. So he had to get his skid steer in there and had to lariat him tight to it and then get a nosebleed in it. And then I had to tube him to get my, now I lost the trade name of it. It's going to be in the next picture. No, it won't be in the next picture, the picture after. These are the normal beef cows. We had 2 beef cow herds this spring. The cows are fine, but here's the calf. A calf that's laying down was born kind of in a coma, and he's been down for 8 days. And the other one is the first day down, can't stand up. I saw it slobbering and running its mouth funny. And here, there's the treatment, lactulose. That better looking calf, the 5-day-old calf, I gave it 15, well, I gave it about 20 cc's because it can't swallow good and I didn't want to tube him. Anyway, I gave that to him 4 hours before he's standing up chasing the cow again. But since the cows are kicking that out in the milk, 2 days later we had to treat him again. And 2 days later we had to treat him another time. And then these people were such good operators as dairy people, they never had scours with their dairy calves, and then this is the first time we ran into this in their beef herd, so we were all tearing our hair out. And what's sad is, my partner and I overlooked the botulism at first because we thought they were just, it was in May, we thought they were just turning them out on pasture, and then we thought they were just eating dry hay. Well, here they had a upright silo on another farm that had, that's the sample of my truck in a jar, the butyric acid smelling haylage that's not Roundup. They were hauling that up and feeding them 3.5 pounds of that a day to the cows. So, anyway, we ended up saving most of them. And it was this lactulose. That lactulose is a man-made sugar that Monica Kruger told me to use in 2014. It's a synthetic sugar that isn't absorbed. So it's like aspartame, the sweetener, see, that isn't absorbed. So you're supposed to, stuff is supposed to taste sweet, but it ain't gonna make you fat or hurt you. So somehow this lactulose, it's made for, you can't read the label, but it's made for humans with botulism. That's what it says right on the label. Give 15 to 30 cc's to an adult with botulism. And I bought it from our veterinary supplier as soon as I came back from that meeting in 2014. And the damn stuff was outdated in 1st of 2015. But I still had some quarts around, thank God, so we treated the stuff and it works. It still works, thank God. But if you don't have that, and I wish I'd done this to the bowl, sauerkraut juice. Cabbage, botulism is a cousin to tetanus, but tetanus works on sensory neurons and botulism works on the neuromuscular junction. Acetylcholine is the neurotransmitter, and cabbage is the only vegetable we have that kicks out a lot of acetylcholine, and it's a Lactobacillus plantarum that makes sauerkraut. I'm pretty sure that's the right bug. It's one of the few bugs on the planet that'll break down glyphosate. So for that bowl, I ran down a dairy plant that makes his own sauerkraut, and then I strained some of it and went over there and gave him that. And we kept him alive we had to keep him for 30 days because you got to treat him with a penicillin drug, and he got aspiration pneumonia. See, they can't swallow. They got to drink like a cat, and they raise their head, and then some cud runs down in their lungs. So what's really sad is the dairy herds where we really had trouble with this. Here's a surviving cow on one of the 2 dairies where we had this real bad. See, I was fighting this back in 2014, and it's been happening a lot since. Well, most of the cows don't get this bad. What they do is they're just kicking out enough of that neurotoxin that they got to drink like cats. So a whole bunch of these dairymen think they got stray voltage, even in the summer, even when the element's turned off, because a cow, a dairy cow needs to stick her nose in there and drink 25, 30 gallons down. Well, instead they're drinking like cats. So then I get in there and second guess with the feed man, and it don't matter, even if we make pretty good quality feed, if you're not there at 6 months before when they fed the bad silage, it was too wet. It looks like I had Ames come out and second guess me, and they said, well, that guy makes the best feed around. And well, he did then, but they weren't there when you could smell the haylage he made 6 months before to get this thing started. And so what happens is they don't eat enough, and if they don't, water's your most important nutrient. If they don't drink enough, then you can't make them eat enough. So big Holstein cows that should be eating 55 pounds, you couldn't make them eat over 45 pounds. Then you're shooting with XNL or Exceed, those are second generation penicillins, so you knock down the botulism, you sell them. So we got dairies right and left that are still seeing this. We don't see it so much in dairies anymore because we don't have any left. You know, they're quitting.
That'll cure.
Mycotoxin issues. Well, corn byproducts at ethanol plants have a 3x more mycotoxin than the corn delivered to the plant. And that's because the germ is what makes the ethanol, and your mold's always growing in the hole on the outside. And Dr. Bob Kremer, one of my other mentors that worked at Fort Detrick, with Huber and McNeil, or after them, they all have that common denominator. He worked for our USDA, and he got totally run over. This glyphosate did help us get rid of the moldboard plow, so all the USDA head honchos above him bought into that. So most of his stuff is published in Europe, in the European ag journals, and he did some work just south of Bruce at Earlville on one of the farms. We're going to wind up to do some testing there. So he tested some bean ground 3.5 years after the last glyphosate was used because we changed tenants, and it still had over 3 quarts per acre. And see, in my land grant colleges, I got I think Wallace's farmer back there where the land grant college guy's still saying there's no soil carryover with glyphosate or glufosinate either one. And it's just it's a lie. I mean, it's because in clay, yeah, actually we're sort of lucky in northeast Iowa with that sandy loam soil because we still had 3 quarts per acre dose 3.5 years after it was last used, even with what ran down the Mississippi and with what went into our well water. And you can have good black dirt and clay where the half-life of the damn stuff is 22 years. So the last thing we should have done is just make, the USDA and the organic people got together and they said, we can't use NPK. So we should be able to use conventional manure from our neighbor. And see, if you use turkey manure or hen laying manure where you fed Roundup grain, or if you use hog manure, and especially if you're putting on 6,000 gallons per acre, you're dosing the hell out of your field with glyphosate. So then you see you gotta, so that's why if you're working with Merlin or these guys, you gotta keep putting the stuff on because it gets knocked down again. Glyphosate promotes the fusarium mold. So just because you get more mold doesn't mean you're going to have more mycotoxin, but if you get the weather with it, you sure will. So it makes me so mad when, Miriam Hernandez up in Canada was able to show that there's a better correlation with wheat scab, which is another thing sort of related to our fusarium problem in corn. She could show a better correlation with whether you were going to have that with how much glyphosate you'd used the previous 2 years than what the weather patterns were. And Kramer showed the same stuff. So those heifers are virgin heifers. And they just got enough zearalenone, so some of the stupid prolapsin, in our feeder cattle when we're feeding so much either gluten or distillers or whatever. It varies according to year, according to rainfall or whatever, but we're promoting this family. Yeah. It doesn't matter if it's Alltech or who it is doing their 37 plus, they're all telling us that this problem just keeps getting worse. So actually Michael McNeil, I, and the late Bob Straight, because Bob worked for the president of the Iowa Corn Growers, we got asked to go down there and talk to Wendy Winterstein and a whole bunch of the farmers. There was a closed door meeting at Johnston, and we got asked to go down there and talk about this issue. And we tried to rile them up and have them look into it. And they had a meeting after, I ran after Wendy and these guys and I said, well, what do you think? Because we talked in the morning and a couple of young bucks. See, it's always younger and younger people that come to debate Huber and I and the other people if you debate them. It's never the old guys at the head of the company because I think a lot of times they think, hey, maybe those guys know what they're talking about and they quit. and then you send out the new guys that you got their scholarship money from them, and you go through the same thing. Well, anyway, the whole thing got tabled. Well, I left my stupid little computer there and started driving back home on the interstate, turned around, went back, and here are these two young bucks from Monsanto up in the corner talking to Dr. Michael McNeil yet about, hey, if you guys are wrong, or if you guys are right, what's our liability? They weren't even really concerned about the whole mess that's causing. All they were concerned about is their own liability. And then Wendy and those other guys went in there and they decided just to table it. We'll live with it, you know, or we'll breed up another seed. So Joe Hall, Uri Joe Hall just got let go by Purdue University. And that's where Uri Joe Hall was Dr. Huber's first grad student that really helped him work on glyphosate. And he stayed at Purdue after Huber left. And Huber doesn't know why, but anyway, he was working on a flex ear hybrid. See, now we're talking about growing short corn because we'll be able to use drones and spray it better and all this stuff. Well, Uri, if you aren't raising corn silage, so you don't want the stalk and the leaf and the tonnage, he's got a flex-eared hybrid, so he can get, he was up to 6 ears on a stalk that would support itself, and definitely a 2-eared hybrid. They promised when they kicked him out and gave him early retirement. Again, Purdue's another land-grant college. Somebody in there from industry is saying, well, we want to sell just as much seed as we always did. Who in the heck can be against if you got a stock that'll support itself and you can have 2 good ears per stock? Why would you be against that? Well, they confiscated all of it, and this just happened in the last 2 or 3 months. They don't even think he'll get any of his stuff out of there. And he's the one that showed with Huber, you cannot kill a Roundup sensitive weed in irradiated sterile potting soil in a greenhouse. If you protect it, because what you're doing when you shut off the shikimate pathway, that's the pathway that kicks out lignin and phytoalexins and all these what you're doing is you're shutting down the plant's immune system. That's why it doesn't dry up and die like 2,4-D, because you're not speeding up its metabolism. You're using pathogens to kill it. And the reason these water hemp and some of this stuff is so resistant to it, that's what grew down in a creek bed or whatever, and see, it can tolerate fungus better. So it seems kind of crazy that we spent 50 years using something that promotes disease to be our main herbicide. You know where I stand. Okay, many unanswered questions. Dr. Tom Swirzysek, Mike Sheridan and I, we actually, and Mark Curley, he grew up in Ryan, we mentored him, and then he became the head honcho of the National Animal Disease Lab for years at Ames. Well, now he's interested in this stuff now that he retired. But I tried to work on him before. He was supposed to go, Dr. Tom Swirzysek, if you don't know that name, he is the veterinarian that posted Secretariat. And his brother Steve Swirzysek has an organic herd in the middle of, he grew up in the middle of Nebraska and had a cow herd. I don't know if he still has it going or not, but Dr. Tom has passed away now. But anyway, he's the guy that back in the early 2000s, they had a real early spring. And with these racehorses, they give them artificial light and all that stuff because they try to have their foal as close to January 1st as they can, because they want it to be big when they're racing either a stud colt or a filly or whatever, you know, a year and a half later or whatever. And so that year, what they do is once they're like 2 months along or whatever, and end of February, March, they ultrasound them all, and they don't insure the mare that's the mother of them, they insure the unborn colt for millions and millions of dollars. And so in the early 2000s, not too many guys are interested in equine around here, but this mare infertility syndrome, and it's still here. What they thought happened is they called it redworm poisoning, because there are all sorts of wild cherry trees down there. They had an early spring, they turned them out on pasture. After after doing the ultrasound on them, and then then they had a warm spell, they turned them out in a pasture, and then they had a cold freeze in April. So it killed all these webworms on the cherry trees, and the mayors, they thought, ate those webworms and that aborted them. Well, the way the insurance policy was written is we won't pay you unless you got a veterinary diagnosis. Well, good old Tom, because he was using his electron microscope and actually knew something, and was working with this agent, that a Biomatrix or a Prion, we still haven't had anybody to really follow up. And of course, the veterinarians I tried to work on that know nothing about it, they all said, oh, he's just looking at an artifact. Well, he wasn't looking at an artifact. Anyway, he held up, I don't know how many millions of dollars of insurance money, and so then they fired him as the director of their diagnostic lab, even though clear up into his early 80s, he's the one that had to do all the damn drug testing at the Preakness and the Belmont Stakes and the Kentucky Derby. So, anyway, so there's other things. So, we need to shut up. That's a grandson, and that's a cat bed out. To keep her frog legged, I had to have the client putt and go ahead, and then my grandson's sitting on it just to keep her frog legged. She made it, but entertaining. And here's some books. That Breakfast at Monsanto's, that's written by this medical doctor that lives on, is it Kauai? Is that the island where the seed corn companies make their seed in case they have a disaster on the continent? Anyway, that son of a gun, for 2.5 years, he got put on a citizens panel to debate industry and go through all the science supposedly on glyphosate, because it's actually, it's more dangerous in salt water than it is in fresh water. So where it's running off, it's killing their coral reef and doing all sorts of shit there. Well, anyway, when this thing came to a head, when he got done at 2.5 years, he was the only medical doctor on the committee. Anyway, it came out against industry, but 55 to 60% of the people on that island are either directly employed or indirectly employed by the seed corn companies. So they went and talked to them, and of course, everybody voted do nothing, because again, it was going to ruin their economy. So then this guy's wife, and he's a decent guy. The only problem with his book, in my opinion, because the proof is all in there, he says, is Roundup in our food making us fatter, sicker, and sadder? He doesn't need the question mark. But his wife says, you got to write the book, because see, they got run over. In other words, they spent all that time, and then even though the whole thing, the industry guys quit the last day because they knew it was going to come out negative, and then they just tabled it and said the hell with it. And the other book was written by my daughter Leah, that's teaching science out of Holyoke Junior College. But anyway, I was hoping it was outdated by now, but it isn't. It's still appropriate. And there's I need to show that chart that Terry Fromm held anyway, so you can take a picture of that page. The synthetic pesticide use in Africa, we want people to read that here, but a bunch of us donated our time and everything to write that textbook, because Huber or Charles Wilson worked on us. See, there's been tremendous pressure lately. Big agriculture, or Africa never really joined our green revolution. And so they're trying to say, you ought to copy everything we did. And so some of us are saying, look at it, everything we did isn't necessarily right. So I think that's, it didn't just start Dunham's chapter in there, there's some good reading there. And then that book, Mineral Nutrition and Plant Disease, that was one of the books, Huber, when I first met him in 2009 at an organic conference, he gave me my copy. And it's sort of beat up now because it's been in my vet truck. That's another thing that's really helped me on this molybdenum and cobalt thing, because you realize if you dig up in June or July, go out and dig up some soybeans, if you got alfalfa, rig up your alfalfa, now the nodules are real small, so you might have to borrow a scalpel blade from the vet before you could use a pocket knife. But anyway, split that nodule. There's pictures in that book to show you what it ought to look like. If you split the nodule, and you gotta have that in every legume, the rhizobia bacterium in there, or whatever fixing nitrogen. If they don't have molly, that's a cofactor for nitrate reductase. They can't fix nitrogen. And so we got all sorts of beans, we still have the protein and stuff in them, but then we gotta put on more nitrogen in our next crop. It isn't producing the nitrogen it should. Where you cut that nodule in half, it'll be either lily white or pea green. If it's fixing nitrogen, like urea, it ought to be pinkish brown, and it will be. And I've got numerous clients now that Bob Street had a lot of connections with a farmer down by Stockton, and then a guy at Medora, Iowa that I've worked with, so they've been bringing up some foliars to try to take care of that, or that seed treatment. This is the outfit that's paying for our extra 2-day meeting, Agriculture Beyond Glyphosate. Saga of the soggy sauerkraut. And you probably, I don't know if you can see that, you can sip it. But anyway, Bear Creek, Wisconsin. Michael Maney is the CEO of it. CLK Sauerkraut is the biggest commercial sauerkraut company in the country. And what happened to them is, see, everybody wants to eat organic. So they wanted to produce more sauerkraut organically. Well, what happened is, our USDA got in there and the organic people got together and said, well, we can use sauerkraut, we can use manure instead of NPK, we can use manure from a conventional guy. And so it was turkey manure up there, because they're halfway up Wisconsin, didn't have a hog. So they used that. They lost over $1 million for it. So now see, they're paying for us to speak, and then they've got a use for the throwaway sauerkraut juice. There's some big dairies that have put in a huge bladder, and then this guy will truck it down on an old milk tanker truck. What do they hold? 6,500 gallon or something like that, I think. And he'll fill that bladder up, and then see, they'll meter that into a TMR. It doesn't work good component feeding a small dairy because they don't like the taste of it. And it was also bringing it down because they found out if you put that on in the fall and scratch it in a little or whatever, your old the planetarium, the Lactobacillus plantarum, will break down the glyphosate. So by the next spring, you actually truly have a field that you could plant non-treated corn on immediately and not have a little yield loss. And Martha Carlin now, she's listed on there. They use some of her stuff. She's a real sharp thinker that came from industry. She's one of these systems analysis persons, and she lost her husband with Parkinson's disease, and she got fed up with the human medical system. And so she got together with this Raul Kano, a professor that's been all over the world, getting old bugs from Antarctica and volcanoes and all over, and so they got together, and so they've got a concoction that you can put on that'll break down glyphosate. So anyway, yeah, issues. Oh, and this is something Howard Vligler just sent me. This is a study from British Columbia, from med schools. It's in peer-reviewed medical literature, and that'll show you how, see, all our EPA never did any science at all, and they don't ask industry to really do science. And what they do, though, they're only worried about acute poisoning. They've never looked at chronic poisoning. So they've never fed pregnant rats, pregnant mice, pregnant rabbits, and then find what happens to the F2 and the F3. And this study, by God, I don't know any of these doctors, but Howard found it, and I don't know who sent it to him, but I think it was done and published. They used crowdfunding to do it, Because see, if you try to go to land grant college, you can't find anybody that wants to ask these questions. So they crowdfunded and got it done. That thing is worth reading because it fits in with what that medical doctor has said. When you go to the F2, and rats, mice, and rabbits don't have that long a generation time. We could have paid to do that. And then, guess what else too? We use Purina rat chow to feed them. And Purina doesn't go out and buy, they don't go out and buy organic corn, to make the rat chow. So a lot of their studies are screwed up there. But they sorted through all that. Well, anyway, the dementia, these animals can't even go through a maze. It's causing genetic changes that show up in future generations, more so in ours, and more so than in the first. That's it. So, I'm still passionate, if you can tell.
Thank you, Art. Mike? One of my farmer friends is here, Monty Casper. Monty's got a horror story to tell you about fungicide. Monty's been with us on humates and carbon on the soil and these livestock. And I think I can step out here and basically say very freely, Monty does not lose. He and his son milk about 100 cows. And he's told me before many times, once we get a baby calf on the ground, 10 days, we don't lose a calf unless it's injured. Monty, would you tell the rest of your story?
Well, here, what was it? Probably 8, 9 years ago, neighbor thought he had a helicopter, put fungicide on. Across our buildings, you could wipe your face off. Week later, we lost 11 peppers out of 11. All respiratory. We've been using the humates now since
5 years already, I think.
At least
6, 7.
just shortly after. When did you start working with?
Well, it's been 9 years already, I think.
We started shortly after
I
that.
started in 2015.
Right after that, we put it on, we put 200 pounds the acre on every other year, and we feed the cows. And,
Reed says Pete.
yeah, right. And,
Then the AFCO road.
we've had the vet on the farm once a year since.
Monty's probably the most profitable farmer that I know we work with.
And the only reason we have them on once a year is so we get our medications for our calves. Because you gotta have them on the farm once a year to get prescription. Otherwise, we don't have problems. We've grown our herd in the last 5 years from 65 cows, we went up to 130 tonight, and we've got 0.
I can do that.
We don't do sex semen. It's just the cows don't.
Burned out.
Well, they don't. We just gotta get rid of them because we got too many.
The Iowa State vet program is gonna have to shut down if more guys do that, you know that.
Well, you're just helping out with the shortage of large-animal pets.
Well, we're running right now, we run a mixed herd. And we're running a 5% butter fat tank, averaging a 3.6 protein.
You're gonna take a Casper test.
I fed no protein, no purchased protein since we started humates. The cows are healthy. I don't know if you know anything about hairy warts. In the feed, we have an average of 1 to 2 a year. We don't treat them, we don't.
Usually, once you get hairy warts on the farm, it's 85
I mean, usually it's the hoof trimmers here every month, fixing 6, 8, 10, 2 a year. And we don't even treat them. They cure themselves. That guy's a lifesaver.
I want to thank everybody.
God,
This start went long. We'll have a video for this and I'll email it to you. It'll probably take a good month time to get some editing all this stuff.
We have to sign up so you get our emails, so if you have it.
Well, or just call me. We'll get you connected. I'm going to tell you some more stories as soon as Terry shuts the camera off.